Polysaccharides. Objectives. How are alpha-glucose molecules linked to form starch? What is a chemical test for reducing and non-reducing sugars? What is a chemical test for starch? Polysaccharides are polymers. So monosaccharides join together by condensation reactions to form glycosidic bonds. Polysaccharides are large and insoluble, and they are often used as storage molecules. They are hydrolyzed disaccharides and monosaccharides. They could be used for support, like cellulose in plants. As an example, starch, which is a storage molecule, is found in starch grains, and it's 200 to 100,000 alpha-glucose molecules long, and it forms a very complex shape. So, to form starch, it is a polymerization reaction by condensation reactions, and starch is very much a storage molecule. It contains two types of subunits. It's got amylose, which has 1,4 glycosidic bonds and forms a very compact helical structure, and amylopectin, which has 1,4 glycosidic bonds and 1,6 glycosidic bonds, and it is highly branched. Cellulose is made from beta-glucose molecules, and because of that, the hydrogen bonds form cross bridges between the parallel layers of beta-glucose chains. This gives it enormous strength. So, what we want to know is, how are alpha-glucose monomers arranged to form the polymers starch and glycogen? How are beta-glucose monomers arranged to form the polymer cellulose? And how do the molecular structures of starch, glycogen and cellulose relate to their function? So there are many different molecules with many different properties, which can all be made from a small range of smaller molecules. Larger molecules are different because of the way smaller molecules are combined, like in polysaccharides. So as a recap, this is the structure of alpha-glucose. It is a hexose sugar, and it has got 1, 2, 3, 4, 5, 6 carbons. On the carbon 1, the hydrogen faces upwards and the OH faces downwards. However, on beta-glucose, the OH and the H are flipped on the carbon 1. During a condensation reaction of two alpha-glucose molecules, an OH is taken from one of the alpha-glucoses and a H from the other to form H2O. So it's a condensation reaction because water is removed or released, and therefore you get a glycosidic bond, and the disaccharide that you form is maltose. So now looking at the polysaccharide starch, which is found in plants. So starch is a polysaccharide which is found in plants in small grains. And there is large amounts of starch in seeds and storage organs like potato tubers. And it is the major energy source or food in our diet. So starch is made up of chains of alpha-glucose linked by glycosidic bonds formed by condensation reactions. It is mainly an unbranched chain wound into a tight coil, which makes the molecule very compact, and it stains blue-black with iodine solution. Now, starch's function is that it's an energy storage, and it's suited for this because it's insoluble, it doesn't draw water into the cell by osmosis, and it doesn't diffuse out of the cells. It doesn't affect the water potential. It is compact, so lots of energy is stored in a small space, and it is hydrolyzed to form alpha-glucose, which is easily transported and used in respiration. Now we're looking at glycogen in animals. It's got a similar structure to starch, but the shorter chains It's made up of shorter chains and it's very highly branched. It's called animal starch because it's the major carbohydrate storage of animals. It's stored as small granules in muscles and liver. The structure for storage is for the same reasons as starch, but because it's smaller, the chains are even easier to hydrolyze to alpha glucose. So now we're going to look at the structure of cellulose found in plant cell walls. And cellulose is formed by the condensation of beta glucose to form cellulose. And cellulose is formed when beta glucose is linked by condensation reactions. But you can see that you have to flip every alternate beta glucose to form the bond, the glycosidic bond. So every beta glucose is flipped, and that means you have OH group facing downwards and facing upwards. So when they then form layers, you can get hydrogen bonds which form between these hydroxyl groups. So cellulose differs from starch and glycogen in one major way. It's made of beta glucose. This small variation produces fundamental differences in the structure and function. In beta glucose, the hydroxyl group is above rather than below the ring, meaning to form glycosidic links, each beta glucose must be rotated by 180 degrees compared to its neighbour. So the CH2OH group on each beta glucose alternates between being above and below the ring. Cellulose has straight, unbranched chains that run parallel to each other, allowing hydrogen bonds to form cross-linkages between adjacent chains. Many hydrogen bonds strengthens the cellulose molecule, which means it's a good structural material. So now if you look at cellulose's function, cellulose molecules are grouped together to form microfibrils, which are arranged in parallel groups called fibers. Cellulose is found in plant cell walls and provides rigidity. It prevents the cell from bursting as water enters by osmosis by exerting inward pressure to stop the influx of water. Plant cells become turgid and push against each other to make stems and leaves semi-rigid to provide the maximum surface area for photosynthesis. So what you really need to know. You need to know that polysaccharides are formed by the condensation of many glucose units. Glycogen and starch are formed by the condensation of alpha glucose. Cellulose is formed by the condensation of beta glucose. You need to know the basic structure and functions of glycogen, starch and cellulose. And you need to know the relationship of the structure to the function of these substances in animal and plant cells.